After the mitosis process, the meiosis comes into place. The meiosis is a special process of reductional cell division. Spermatogenesis is a process of sperm cell development. It is rounded, immature sperm cells change into spermatoza. Prophase 1 is the process wherein the chromatin is condensed because of lepsonin. Then, the homologous are paired afterwards. Then, pachytene, wherein the crossing over and recombination happens. And then, the diplotin, wherein complexes dissociate. And lastly, the diakinesis, wherein the chiasmata disappear and homolog centrome binds into a spindle fiber. In meiosis 2, cell moves from meiosis 1 to meiosis 2 without copying their DNA. During prophase 2, chromosomes condense and the nuclear envelope breaks down if needed. The strand to centrosomes moves apart. The spindle forms between them, and the spindle's microtubules begins to capture chromosomes. During metaphase 1, homolog pairs, not individual chromosomes, line up at the metaphase plate for separation. In metaphase 2, the two sister chromatids of each chromosomes are captured by microtubules from opposite spindle poles. The chromosomes line up individually along the metaphase plate. The anaphase 1 is the homologous are pulled apart and move apart to opposite ends of the cell. The sister chromatids of each chromosomes, however, remain attached to one another. In anaphase 2, the sister chromatids separate and are pulled towards opposite poles of the cell. Finally, in the first part, the telophase 1, the chromosomes arrive at opposite poles of the cell. In some organisms, the nuclear membrane reforms and the chromosomes decondense. In telophase 2, nuclear membranes from around each set of chromosomes and the chromosomes decondensed. Cytokinesis splits at the chromosome sets into new cells, forming the final products of meiosis. In this video, we will relate the process of oogenesis towards a quantitative analysis of political data for students of political science. The first stage occurs when a singular primordial germ cell undergoes mitosis to produce two diploid oogonia. This phenomenon is an example of linear relationship in the quantitative analysis of political data. A linear relationship occurs when an increase in the independent variable, the singular primordial germ cell undergoing mitosis, leads to a constant increase or decrease in the dependent variable, the two diploid oogonia. The second stage occurs when each oogonia also undergoes mitosis to produce two diploid primary oocytes. This phenomenon is an example of a negative relationship in the quantitative analysis of political data. A negative relationship occurs when the lower value of the independent variable, the singular oogonia undergoing mitosis, leads to an increase between the dependent variable, the two diploid primary oocytes. Occurs when the primary oocyte completes its first meiotic division, producing a secondary oocyte in the first polar body. After that, the secondary oocyte enters meiosis 2 and is stopped at meiosis 2. In addition to the occurrence of ovulation, at this stage, once the meiotic division has been completed and a secondary oocyte has formed within the dominant follicle, there is also a 50-fold increase in the oocyte. This phenomenon is an example of an additive relationship in the quantitative analysis of political data. Additive relationship occurs when the control variable, the dominant follicle, is held constant. The relationship between the independent variable, the primary oocyte, and the dependent variable, the secondary oocyte, strengthens. The third stage occurs when each primary oocyte starts to undergo meiosis 1 to replicate their DNA, but they are then halted at the first meiotic prophase. This phenomenon is an example of a spurious relationship in the quantitative analysis of political data. A spurious analysis or relationship occurs when after the control variable, the meiotic prophase, is held constant, the relationship between the independent variable or the primary octet and the dependent variable, the DNA replication, weakens. One stage occurs when the secondary oocyte completes the meiosis II, which produces the mature haploid gamete, the ova. This phenomenon is an example of zero-order relationship. In a zero-order relationship, there is an observation between the relationship of the independent variable, which is the secondary oocyte, and the dependent variable, or the ova, without taking into account a rival explanation.